All right, so we're gonna finish up the cardiovascular system. Should realize for completeness, when we were talking about vessels, I didn't mention a couple of the common kind of pathologies of vessels, which you should at least know them by name and the basics of what they are. What is an aneurysm?
Isn't it like a bulging of the blood vessel?
Yeah, exactly. So it's when there's like kind of a weakening of the vessel wall, so then the pressure is able to kind of balloon it out. So this is the blood vessel. There's like some part that's actually kind of ballooning out here. Because it's a weakening of the wall. This obviously, just in and of itself, is kind of bad because it's going to be putting like pressure on surrounding structures. But what's more problematic is it's a potential for just a complete bursting of the vessel at that place where it's weakened. In which case you get a burst aneurysm, in which case you got blood pouring out. Like, so this could be like a hemorrhagic stroke or something where you are now no longer getting the blood going to where it's going, should be going, and instead just pouring out. If it's a small thing, it might be just, you might recover from it. A large, a major cerebral artery, or like people have aortas that have an aneurysm in their aorta, and then their aorta bursts, and basically within a half a minute you've pretty much poured all the blood out of your cardiovascular system. So a burst aneurysm is really dangerous. If they see that you have an aneurysm, they'll monitor it, or if it looks bad, they'll go in. There's different ways to try to stabilize the vessel so it won't explode or burst on you. So aneurysms, atherosclerosis is another sclerosis, hardening of the arteries. This is when you have your blood vessels and you start getting these things, they're called plaques. It's like kind of the smooth muscle starts proliferating, you get lipids in there, and it basically starts to narrow the passage, of the lumen. So this thing here, we call this a plaque. It's basically smooth muscle and lipids. And that's obviously going to impede blood flow, increase resistance, make it harder for the blood to get where it needs to go. If this is happening in your coronary arteries, it means that your heart is getting less and less of the blood it needs to do its job. If this is in your carotid arteries, it means your brain isn't getting enough all the blood and oxygen it needs. And it happens. It is related to diet. It's related to saturated fats and things like that. But it's not about like the fats actually coating the inside of your vessels. It's more about this more complicated inflammatory response that ultimately results in the formation of these plaques. These plaques are in the wall. I think sometimes the way people kind of casually talk about, fat blocking your arteries, it sounds almost like the fat is sticking to the inside of the vessel, but it's actually this structure, this plaque growing within the wall of the vessel that is the problem. And again, like I said, is correlated with, kind of diets high in saturated fats and all that, but in an indirect way, not like the fat's gumming up your arteries or something. So anyway, these are just, it'd probably be silly to leave vessels without at least mentioning these. We're talking about lymphatic system. We talked about a lot of different things it's doing. I realized just as a teaser, I should just introduce this idea. This is also going to be critical for absorbing fats from your diet. And this is because fats don't dissolve in water, as you know. So instead, all of your fats are going to be constructed into these lipoprotein things. We're going to see the chylomicrons, but then those are too big to absorb into a capillary. So instead, you're going to have a lymph capillary, specific one we're going to call the lacteal, which ultimately is going to, where do all the lymph capillaries eventually dump back into?
Vena cava?
Yeah, exactly. It's going to dump back into the brachiocephalic vein, into the vena cava, and just join into your bloodstream. So this little lipoprotein thing that is the way we're absorbing fats from our intestines will eventually end up in your bloodstream, but it will get there in a roundabout way. Rather than getting absorbed into the capillaries and then going through the hepatic portal system to the liver, it's just going to go straight through the lymphatic system and get dumped straight into your circulation, your venous circulation. So we'll come back and visit that later, but I figured I should just add a little teaser in there so it's not going to seem like a surprise a little later. Okay. So we're going to finish up talking a little more about blood. Again, we've already talked a lot about blood, so we're just going to go into a few of the points in a little more detail. Talked about plasma versus the formed elements. Again, we said, what's a typical hematocrit? What's a typical proportion of formed elements like cells and such to the plasma?
55% plasma and 45 formed elements.
Yeah, that's a classic, and again, we saw it can be different. It tends to be lower for women than for men, but it's about right. And let's focus on the plasma in a little more detail now. Plasma obviously is mainly water. So, again, these numbers are always a little silly, but I'll say 92% H2O, maybe 1% dissolved solutes. You got glucose, etc. Gases, whatever. So, and right, adding in some sugar doesn't change the volume a lot, right? If I take a cup of coffee and I put in a heaping spoon of sugar, it doesn't change the volume much. So all of the dissolved stuff in the plasma, adds like about another 1% to the overall volume. And then that leaves us with 7%, and these are going to be the plasma proteins. And I will kind of just introduce the basic, you've already met some of them, like angiotensinogen was sitting in the plasma as a protein ready to go for the renin-angiotensin mechanism. Fibrinogen was sitting there waiting in the wings, waiting for the blood clotting to happen. There are so many different plasma proteins playing so many different roles. The most common of the plasma proteins is albumin. think it's like 60% or something of the plasma proteins. Um, One of the big things that this does is it helps preserve osmotic balance, right? We need to make sure that the plasma is 0.3 osmolar, or else we know what happens to blood cells. And we also know what's going to happen. The plasma is going to leak out of the vessels into the tissues, or water will get sucked out of the tissues into the blood vessels. So it's important that our plasma is 0.3 osmolar. And this albumin protein helps balance the osmolarity of the plasma to make sure that it hits that target so we don't have water moving someplace we don't want it to move. There are also like alpha and beta globulins. And these act as carriers. So, and not just these, actually albumin can act as a carrier. There's lots of other specialized proteins that act as carriers, but these are for molecules that don't dissolve in water. Carriers, for lipophilic molecules. Right? If you have a steroid hormone can't dissolve in water because it's fat-based and it's hydrophobic. So you have to have it bound onto something that does dissolve in water to actually transport it around in the bloodstream. So these globulins help bind things that normally wouldn't dissolve in the plasma, so you can actually transport them in the bloodstream. Lots more. Like I said, all the proteins involved in blood clotting, of which there are tons, a whole bunch of things involved in body defenses, which are tons. I shouldn't say tons, lots. Literally tons. No, I'm just kidding. So these are all made in the liver. So when we later on are talking about all the things the liver does, this is just one of the many things the liver does, is it makes your plasma proteins. Right. And this, like, if you ever see like the little kids who are starving, kwashiorkor, like people who don't have enough food, all they eat is like grain, you can actually end up getting, they have big bloated bellies, because you're not being able, if you don't have enough amino acids to make all of the plasma proteins properly, then the water isn't going to stay and leaks out of your cardiovascular system and into your abdominal cavity and stuff like that. So there's one other plasma protein that's not made by the liver, which we'll talk about in way more detail later. And we've already introduced the antibodies or gamma globulins. These are made by lymphocytes. But don't worry, like, we're going to talk way more about them. But just for completeness, just saying that there is one class of protein that's floating around in your plasma that is not made by the liver, but is one exception. It's one exception to everything else. So I think that's what I'm going to say about plasma. Plasma, mainly water, dissolved solutes, these plasma proteins, and the plasma proteins help preserve osmotic balance. Some of them are important for carrying around lipophilic molecules. A lot of them are doing all sorts of things. Again, part of blood clotting, part of immune response, part of blood pressure control. So lots of things. And one of the things that's always a little like non-intuitive, like all those bacteria in your gut, they're making stuff too. and that actually gets absorbed and gets transported in your blood. So your blood is actually transporting stuff that's actually made by your bacteria in your gut that actually seems to influence you. There's like some evidence that it interacts with your nervous system, like your serotonergic pathways don't develop properly if you don't have the appropriate bacteria making stuff into your body. Supposedly, what kind of foods you're drawn towards can be influenced by the bacteria in your gut. Like they release things that go through your bloodstream and that somehow affect what kind of foods you want to eat, and put you more towards eating the kinds of foods that are going to provide the substrates that are optimal for those bacteria. So you're like getting played a little bit. Um, anyway, plasma. So formed elements, like 99% are going to be the RBCs, red blood cells, which we'll talk about in some detail. Then there's going to be the white blood cells. These we'll talk about when we get to the immune system. These are immune function. And they are not confined to the closed circulatory system. We'll talk about the white blood cells in more detail later. The lymphocytes, the macrophages, the neutrophils, the basophils, the eosinophils, things like that. So for right now, you should know white blood cells are called leukocytes as well. You should know that they're involved with immunity. You should know that they're not just found in the bloodstream. They're all over the place in your tissue spaces, traveling around in the lymphatic system. They're even like in the outer layers of your skin. They're all over the place. So we will return and see these in more detail later. And then there's platelets, which are going to be important for hemostasis, for blood clotting. And they're not really cells. They're like little packets of stuff that are going to be released in response to a tear in the blood vessel that are going to start the whole process of stopping the bleeding. So what I'm going to do, we're going to spend a few moments just looking at the red blood cells in a little more detail, because you should understand them a little better. And they're going to come up also when we get to the respiratory system as well, which is about delivering and picking up oxygen and delivering and picking up CO2. And then we'll talk about the platelets and the whole process of what happens when you get tear in your vessel. And then we'll be done. So RBCs, aka erythrocyte, erythro red cell. Sometimes it's called red blood cells. They are really specialized. Like they get started in your bone marrow and they start looking like a regular cell, but they start making lots and lots and lots and lots and lots of this protein called hemoglobin, which you all know about. And eventually they eject their nucleus before they go into active duty. So you have this cell here. Tends to have this kind of dimple because they eject their nucleus. So it kind of, the cheeks suck in. So there's no nucleus. There are no mitochondria. It's fine because whatever minimal metabolic processes they got going on, they can do with glycolysis. But without a nucleus, it means that they can't repair or maintain themselves, right? Because that's the blueprint for the, making new parts and pieces. So these things only live like maximum like 4 months. Then they're basically recycled. Cycled in the spleen and the liver. They are filled with like 250 million molecules of hemoglobin per cell. So they're basically bags of hemoglobin. So let's talk about hemoglobin. It's a protein associated with a iron-containing unit called a heme group. Right, so this is one hemoglobin, right? Remember we talked about that quaternary structure where you have a big protein that's made out of smaller third, tertiary structured subunits. So we have these globin subunits, the heme group, which contain iron. Each heme group can bind an oxygen molecule. The globin subunit actually has a place that can bind carbon dioxide. We are going to see when we look at the transport of carbon dioxide, maybe like 30% is being transported bound to the hemoglobin, but to a different site than the oxygen. And again, we're going to look at this in more detail when we get to the respiratory system, but basically the amount of CO2 bound to the hemoglobin actually changes the affinity of the hemoglobin for oxygen, and it will actually cause the hemoglobin to release more or less oxygen under the same conditions in the tissues. So we'll look at the more complicated interrelationship between CO2 and oxygen and binding affinities in hemoglobin a little later. So 4 heme groups means 4 oxygens for each hemoglobin, which means when we go back here, how many oxygen molecules can you put on a single RBC? You gotta think of like Dr. Evil.
About a billion?
Yeah, like 1 billion molecules of hemoglobin. I'm sorry, 1 billion molecules of oxygen bound in a fully saturated hemoglobin molecule. It's fully saturated RBC, I should say. So that's the main thing you need to know about RBCs. Yeah, molecules of so there's 250 million molecules of hemoglobin per cell times 4 oxygen, per hemoglobin equals 1 billion oxygen per cell. I should mention part of the recycling of the hemoglobin, recycling the heme groups creates bilirubin. which becomes part of your bile. It's brownish. This is the stuff that ultimately is going to turn your poop brown. Like you're wondering, why is your poop brown? It's not, it's brown because of this recycling of the heme. If the bilirubin, if there's like liver problems and the bilirubin spills into your bloodstream, that's how you get jaundice. Like your skin and your eyes will start turning like yellow brown if it starts getting just into your general body. And if that happens, you'll also find your poop isn't as brown. I don't know if when I was in grad school, I caught Hep A on a trip down in Mexico and had really bad jaundice, and my eyes were yellow. My pee was just yellow brown, kind of, but my poop was like light gray. That was just the color of the bacteria, which are the main components of your feces. And without having that, the bilirubin was pouring into the rest of the body and turning everything else yellow brown, but the poop was actually staying light gray. It was kind of weird and trippy and super unpleasant. So any questions about red blood cells?
Do you mind just going back to the other slide really quickly? Thank you.
Yeah, so this is a single hemoglobin. Has these 4 globin subunits, again, which have the ability to bind carbon dioxide. These 4 heme subunits, which have the ability to bind oxygen. And then you have like 250 million of these molecules in every single RBC. And again, we'll talk much more about hemoglobin and its affinity for oxygen when we get to the respiratory system on Thursday. Before we leave RBCs, I should also mention one last thing. I know I actually did say, no mitochondria, right? So they're not going to be using up their supply of oxygen that they're supposed to be delivering to the cells. So that's another, part of not having mitochondria there is if they had mitochondria, they'd be kind of sucking down their oxygen that they're supposed to be taking to your tissues. So red blood cell. All right, so we're going to finish up with hemostasis, with what happens when your vessels tear. And again, your vessels are tearing all the time. We saw just now capillaries are literally made out of basically the tissue paper of the body. Like simple squamous epithelium is so thin and so fragile. And the little arterioles and venules are really thin and fragile. Just moving around, things are tearing. Getting bumped, things are tearing. Obviously, more dramatic contusions and whacks and cuts and stuff are going to cause tearing. So there's constantly vessels are getting tears, and you've got to, 2 things. You got to stop the bleeding in the moment so you don't leak out inside. And then in the bigger picture, you got to repair the damage and put it back to normal. So that's going to be these processes in we call hemostasis, hemo blood stasis stopping. And it's going to be both about short-term kind of stopping from, hemorrhaging, and then long-term kind of actually repairing the wall back to its pristine condition. Hemostasis. And hemostasis is mediated by platelets. Sometimes they're called thrombocytes, but I don't want to use that word because it sounds like they're cells. Cyte means cell, and they're not really cells. They are produced, they're made in the bone marrow. There are these big cells called megakaryocytes. And they make these little packets. They just pinch off little bits of membrane filled with granules. So the platelet is basically a little vesicle filled with granules of different stuff.
So the platelets are what you're saying is made in the bone marrow, and the megakaryocytes are in the bone marrow.
Correct.
Okay.
So basically, like here's like your bloodstream. This is actually this would be like a sinusoidal capillary in the bone marrow. God, I am being sloppy as all get out here. Here's a sinusoidal capillary in the bone marrow. Remember, I was saying sinusoidal capillaries were in the bone marrow. These megakaryocytes are here waiting in the wings. And remember, I said sinusoidal capillaries have little breaks in the wall so things can reach in and do stuff. This dude is reaching in and depositing these little packets of stuff that we are now calling a platelet. Like if you are looking at a blood smear, the platelets don't look like cells, right? If you're looking like, oh, look, there's a bunch of red blood cells. Oh, look, there's some white blood cells. There's a neutrophil. Oh, there's a lymphocyte. And then it's like, ooh, what's that little schmutz in between everything? Those are the platelets. You know, they aren't like big cells. They're like little doohickeys floating around in between things. So if you have some blood vessel and there's some tear in the wall, normally the only thing that a platelet should see is endothelium. Right, endothelium, that simple squamous epithelium that lines all your vessels and heart chambers. If there is some kind of a damage, you expose the connective tissue. The main protein that makes like the base layer under the endothelium there is collagen. Collagen is one of the main structural proteins in your connective tissue. So this is going to expose collagen. When there's a rip. And that's going to be the trigger that starts this whole process. So normally the platelets are just bouncing around, doing their thing. So these are my little platelets. I'll make them green here. Platelets. But now they see this collagen. This is going to trigger hemostasis. There are a number of steps to this. So I'm just going to kind of give you step by step what happens when the platelets are exposed to collagen. One of the first things, so that what they start degranulating and releasing granules of stuff. One thing they release is serotonin, which you normally think of as a neurotransmitter in your brain or something. But when they first looked at serotonin, it was actually this thing that was part of blood clotting. It was actually kind of like, yeah, right, it's involved in your brain, whatever. Because it seemed like kind of bizarre, because it seemed like, no, that's for blood clotting. But actually, the platelets release serotonin. Number 5-HT was serotonin. And this causes vascular spasms. So this is going to look like part 1. Platelets release serotonin, which is triggering vascular spasms. Why is triggering vascular spasms going to be a good step here? And spasm meaning kind of constriction.
Limits the amount of flow.
Yeah, it's going to reduce the flow, right? Increasing the resistance, less flow. So we got a hole in the wall. Let's reduce the flow to where the damage is so we don't bleed out as much here. So that's going to be the first step, is release this molecule, which is going to trigger constriction of the vessels, reduce the flow into the area. Part 2, we have what's called the platelet plug forms. So this is basically kind of, yeah, I mean, I don't know if like when I was a kid, everybody started like all jumping on top of somebody else and it's like dog pile. And it's like, boom, everybody's just all on top in a big pile. That's kind of what it's like. All the platelets all start dog piling on each other and basically make a like sandbag. It's kind of like sandbagging. And this is also triggered by other molecules they're releasing. They release ADP actually to attract other platelets to join in the dog pile. So as soon as one starts, they then release molecules that bring other ones into the game. And so this is, again, still kind of a short-term temporary thing of having all these platelets kind of sandbagging over there to keep the blood from leaking out. The next parts that go on are going to be a little more of kind of long-term stuff. So part 3 is going to be what we call clot formation. So this involves lots of steps. There's like over 20 or is it 30 steps? I forget how many steps are. I should probably, let me tell you for sure. Do I have, yeah, over 30 different substances involved. Over 30 steps. This, in the end, I'll give you the last 2 steps. There is a plasma protein called prothrombin. That becomes thrombin. And thrombin is the thing that takes fibrinogen, which we've mentioned, and turns it into fibrin, which is that stringy protein that we talked about already. So after all these 30 steps that happen, you end up with fibrin, which is now kind of more like a Band-Aid. It's this kind of sticky protein that goes and actually puts a temporary seal there. This is kind of more like a Band-Aid. Right, this was kind of just sandbagging, just piling all the platelets on top just for the moment. But now we've got this kind of other protein, this fibrin kind of Band-Aid there. If you have some genetic disorder where any of these plasma proteins involved in clot formation is not functioning right, what do we call that condition?
Is that hemophilia?
Yeah, that's hemophilia. So hemophilia is there's some genetic problem with one of the plasma proteins involved in this whole process, and that means it's going to impair your ability to clot. All right, so now that we've got this we're okay for the moment. We've like put a Band-Aid on it, but obviously in the long term we want to heal it and we want to get rid of the Band-Aid, right? That's what you normally do. You cut yourself. All right, I'm cool for now, but eventually your skin heals and you throw the Band-Aid away. So that's going to be like the, steps 4 and 5. call clot retraction and repair. The fibrin or the clot has actually has some contractile proteins that kind of start squeezing the edges of things together, squeeze out all the plasma, kind of make sure everything's ready to fix. There's what we call PDGF, platelet-derived growth factor. Helps the epithelium and the muscle heal. And so this basically repairs the wall and kind of puts it back to normal. The final part is going to be dissolve the clot. So, once your cut is healed, you can throw your Band-Aid away. So this part involves this stuff, another plasma protein called plasminogen, which becomes plasmin, and plasmin dissolves fibrin. So that's the final step, is you convert plasminogen to plasmin, plasmin dissolves the fibrin, everything's already been healed, so now we're back to where we started, everything's cool. So any questions about any of those steps? So I don't know if anybody's familiar, if you work in an ER or something, does anybody know has anybody heard of TPA?
Yeah, they use it for first onset of stroke in a couple hours, right?
Exactly. So this was like the first actually viable like product made by a biotech company. It was like released by Genentech. And it's tissue plasminogen activator. And the idea is if somebody has a stroke, that's often because there's some blood clot that has broken free and is now blocking some cerebral blood vessel. If you can get them into the hospital within an hour or so, and you give them this tissue plasminogen activator, it's doing this. It's activating their plasminogen into plasmin, and the plasmin is then dissolving fibrin and can dissolve the clot that is causing the stroke. So it's actually a practical thing there. I guess I should mention the idea of a thrombus. Like if I have a clot that starts forming where it shouldn't, that's called a thrombus. It's kind of like an errant clot. And again, that's a really dangerous thing because if it breaks free, it becomes an embolus. Embolus is if it's flowing free, like because eventually it's gonna like get, even if it's not causing problems down here, it eventually, boom, it's gonna clog up and keep the blood from flowing further downstream. So if you had one of these break free and go block the blood flow to part of your brain, you can give them TPA. That will hopefully dissolve that clot and now restore the blood flow. But again, you have to do it fast because if you wait too long, then the brain cells are dead. Thrombuses happen if you don't have adequate blood flow through your veins. Like I said, veins tend to have very low flow because of the low pressure. Just sitting still can increase the chance of thrombuses in the veins. People who have atrial fibrillation, I talked about atrial fibrillation, is when the atria are kind of not doing their pushing well just to move the blood into the ventricles. So the blood tends to just kind of slosh around in there and not move the way it would normally, and it's more likely you get a blood clot forming, in which then if it does break free and goes off into your body, you're in trouble. Now, if it gets stuck in your pulmonary side, it can be a pulmonary embolism and block the flow so your blood can't go pick up oxygen. Or it could be a stroke or a heart attack or who knows what. People who are at an increased risk for thrombus, what kind of meds do you give them?
Anticoagulants.
Yeah, you give them anticoagulants. Which also, again, we tend to call blood thinners. But that would be like that heparin that we saw in that capillary tube to keep the blood from clotting. You know, obviously you need to be careful. If you give them too much, then they're gonna bleed everywhere inside. It'd be like hemophilia. So you have to find the dosage. Like finding the dosage can be tricky. If you give, usually you can tell. Like if somebody is on too high a dose of anticoagulants, they start bruising really easily. Like I think I mentioned, people just get their, ankles get all black and blue just from trying to wear socks. Just the pressure of the socks pushing on their skin and stuff is enough to, have bleeding start that doesn't stop in time because, so, you have to monitor the dosages and all that. But definitely you should know, like, for your exam, there could definitely be some question like describe, hemostasis. There's a variety of different steps, right? The first step is just the platelets seeing that there's collagen. Then they initiate kind of just the constriction to reduce flow to the region. Then they dogpile to make the blood, the sandbag. Then they start the more elaborate, cascade of events to create the fibrin patch. And ultimately there's going to be the, just pull the ends back together, regrow the tissue to heal it so it's back to normal, and then get rid of the clot since you don't need it anymore. And I should mention, it's in your book, so just so you don't get confused. This is kind of the long way to trigger this whole system. There is another shortcut. There's this thing called tissue factor, which is in the connective tissue. So as soon as the blood touches that tissue factor, it will also start clot formation. So clot formation, you can get to clot formation through the steps I've told you right now. But then there's also a shortcut that happens. If the blood just leaks out and comes into contact with this tissue factor in the connective tissue, it will immediately start causing clots or clotting up in there as well. So when you're looking at these pictures in your book and wondering what's going on there, that's what it's that shortcut that can happen to speed up clot formation. Because it's, obviously once the blood is spilling out into your connective tissue, that's a sign like, okay, no matter what else is happening, we got to stop this. Okay.